Hi everyone. So this is our last video that we have this year. So I hope that you have enjoyed the time that we spent together. Although I would have loved to have been in person with you, it has been really nice to be able to record these videos and feel like maybe you were in the classroom with me still. Okay. So our last topic is this idea of polygenetic traits and incomplete dominance and codominance. So what we've been studying so far has been things that are complete dominance, meaning one dominant allele takes over another. So just to kind of recap, when you had two uppercase letters, that meant that that gene was dominant, right? And then if you had an uppercase with a lowercase letter, the uppercase letter kind of hid the lowercase letter and you know, kind of, it was in the DNA, it was in the person, it was in the organism, but the organism didn't show anything for it. But then when you had the two lowercase letters, the two recessive alleles, that showed that trait. So basically what we're trying to get at now is that not all allele combinations work that way. So there's something called polygenetic traits, which poly meaning many, genetic meaning genes. So in this case, you're having many, many genes control a specific trait. So for example, human eye color is actually 3 genes involved. So you can't really do just a simple Punnett square to figure it out because of all the different so with a Punnett square, you're looking at one allele, right? And you're looking at two combinations. But now when you have 3 genes, now you're really exponentially growing how many crosses you would have to do because you would have to try each of the combinations out with each other. Hair color is 4 genes, so that's a lot. But take a look. Weight is actually 6 genes, whereas skin color is 20 genes, while height is 65 genes. So for you to say, "Oh, I'm going to be tall because my father was tall or my mother was tall," yeah, it doesn't really work out that way because there are a lot of different things involved. So you may be extremely tall in your family or extremely short in your family, and your parents aren't tall or short or the opposite of that. And you have to really look to your extended family, like your grandparents and your aunts and your uncles, because all of that information is inside of your parents. So, or a combination of that information at least. So 65 genes, pretty difficult to basically figure it out. So whenever we look at polygenetic traits, we look at like averages. So for those of you that have been to the doctor recently, they always take your height and your weight and they put it on a growth chart. So basically what that chart came about was they collected data from, you know, hundreds of thousands of people, and they came up with what's known as the bell curve, where most of the individuals are located in the middle and average. You have the ones at the one end of the curve that are, like in this case, very tall, and then you have this end of the curve which happens to be very short. So this is what's known as the bell curve, and this is not just for genes. If you ever seen or heard about grades being on a bell curve where the majority of the students, are in the average range, while, you have some students that are above average and do extremely well, while other students just do not do as well. So this bell curve is pretty common throughout a lot of aspects of our life, whether it be through schooling or through height or whatever it is. Okay. And since this requires so many different genes that are actually influencing the trait, you can't use a Punnett square. Okay. So that's polygenetic traits. So the next thing I want to talk about is incomplete dominance, where one trait doesn't dominate over the other. It actually causes a mix. Okay. So here we go. I'm going to cross a white flower and a red flower. So if I could just real quick, okay, here we go. Big R is a red flower with a white flower, which is big W, big W. So it's big R, big W, big R, big W, big R. Move this along. draw. Hold on, move it up here. Big W, big R, big W. So if you take a look in this case, all of them are big R, big W. Now, since neither the red nor the white is dominant, what happens is it mixes. So what happens when you mix red and white? You get a pink color. So that's called incomplete dominance. Okay, so now we're going to take two roses, both with pink flowers. Okay, so we are going to now cross two pink flowers. So let's go ahead. Get my drawing thing out. Here we go. So pink is big R, big W, big R, big W. So let's take a look. We've got big R, we've got big R, big W, we've got big R, big W, we've got big W, big W. Okay, so taking a look at what we see here. I have to do this again. Sorry, guys. What percentage are pink? Pink is the big R, big W. So if there's 2 out of the 4, that would be 50%. What percentage is red? Just one of the boxes, so 25%. And what percentage is white? 25%. So pretty interesting that 2 pink flowers, the percent that the flower that it creates becomes pink is 50%, but there's also a 25% chance that it will be red, as well as a 25% chance of being white. So pretty interesting. Okay. Next, we have this idea of codominance, where in incomplete dominance, they were both recessive, but now we're talking about codominance where they're both dominant and they're both going to be shown and there's going to be no mixing. It looks mixed, but they actually, when you look up real close, you'll see one of each. I'll show you what I mean in a second. So we call that a blending. And that's typical with what we see with horses, okay? So in horses, the chestnut and the white colors are codominant, meaning that they are both capital letters where they're both going to be seen. So when you mate a chestnut horse and a white horse, you get what's called a palomino, where if you take a look at the horse and you get in real close, you'll see a dark brown hair, and then you'll see a white hair, and then a dark brown hair, and then a white hair. So basically what has happened is, since both of those are visible in the actual coat of the horse, it's actually making them look tannish. So although people think that this is a form of mixing, it's not. Where in the example with incomplete dominance, it wasn't a red and a white really close to each other like pigment. It was an actual mixing that caused the pink. Whereas this one, if you look very closely, you'll see some of the hair, is that dark brown color, while some of the hair is a white color. So which causes this like lighter shade. In humans, the codominance example is your blood type, okay? So we talked a little bit about blood type already in the immune system. So in humans, you are going to have A, B, and O. So what happens is when you have A blood, you can have what's known as, let me just move this out of the way again, and up here. Okay, so A blood, you can either be A, because remember you got 2 alleles, or you can have O blood. So people that have A blood, you can either have AA or AO. You both possibilities, you will test as if you have A blood because you have the A protein on it, but those are the 2 possibilities. For B blood, you can either be, you guessed it, BB or BO. AB is only AB. There is no such thing as ABO because remember, your alleles come from your parents. You get one from your mom, one from your dad. There's no three. And then O has to be OO. Okay, so this becomes important when you're trying to figure out possible blood types between family members, if mom is this and dad is this. Okay, so let's just take a look. Excuse me. Oh, it's my daughter's birthday today. Exhausted. Okay, so in a blood transfusion, we talked about this. A blood has to receive from either A or O. B blood is going to have to receive from B or O. AB is going to be able to receive from A, B, or O. Remember, they're the universal recipient, and O can only receive from O, so it's the universal donor. Okay, so A blood, the O is going to sneak in. There's no protein on the O blood, so it doesn't affect that it's there as well. B for the same reason, and then, like I said, AB is the universal acceptor. The O sneaks in. It's got no proteins. It doesn't have a reaction with the body, so everything is good. Okay, so let's take a look. Okay, make a Punnett square of a mom who has AB blood and their dad who has AB blood. Here we go. Okay, so mom is AB. So mom is AB. Dad is AB. Okay, this one is going to be A. This one's going to be A. B, remember, BA and AB, same thing. Okay. So if mom is AB and dad is AB, there is a 25% chance that a child will have AA blood. There is a 50% chance that they will have AB blood. That's 2 out of the 4. And then there's a 25% chance, or 1 out of 4, that the child will have BB blood. Okay. Okay, so here we go. Mom is O, dad is AB. So here we go. Mom is O. Dad is AB. Okay, A, O, A, O, O, B. Okay, so kids are either going to be A, O, or B, O as the possible combination. Okay. Okay, so now if you know a parent has blood type A, but you don't know if they're homozygous or heterozygous, that becomes a problem because then you're talking about, is the parent either AA or AO? So here are two, here's a way to look at it. You are going to have to run 2 Punnett squares. So dad is A, mom is O. So if dad is AA blood, mom is OO blood, you can see that there is only one possibility the children can be. AO, 100% possibility. But now if dad is AO blood, we're going to have 2 possible combinations. Okay, so the kid could either be AO, 50%, 2 of the 4, or they can be OO, 50%, 2 of the 4. Okay, so it does make a difference if dad is either AA or AO, because it is changing the probability of what the child could have. Okay. So now we're going to do the same thing with type B blood, and you can probably predict it will be this. Oh, excuse me. Oh, sorry, guys. Let me just go back. Sorry, guys. Just give me a second. I don't know what happened there. Slideshow. Is this going to let me view from? Makes sense. Start from the beginning. Sorry, guys. Just give me a second. I have such an old version of Microsoft PowerPoint that I'm not able to start it. Okay, here we go. So dad now is B blood, so we don't know if he's BB or BO. So once again, dad is, if it's homozygous, dad is going to be big B, big B. So once again, all combinations of the child are going to be BO, and then dad being heterozygous is going to be BO. Mom is OO. Oops, sorry, guys. Okay, so once again, BO, O, O are your combinations. Okay. Okay, so here we go. Make a Punnett square of a mom who is heterozygous and who dad is heterozygous. So here we go. Mom is heterozygous, so AO. Dad is heterozygous, BO. So we've got AB, BO, AO, and OO. So once again, very interesting. If mom is AO and BO, there are 4 different possibilities that the children can be, which, so if it's 4 possibilities, that means each is 1 out of 4. So you have a 25% chance that the baby is going to have AB blood, 25% chance that they'll be AO, 25% chance that they'll be BO, and 25% that are OO. For those of you, just as a recap, A came down, B went over, B went over, O came down. Doesn't matter if it's OB or BO, same thing, same combination. So it doesn't matter. Okay. Okay, awesome. Let me just. Okay, so we will have an opportunity to meet again this week on a Zoom session. So please, if you did not understand this or need help, also make sure that you have an opportunity to start the lab. I highly suggest it before you come to the Zoom meeting. So if you had any questions, I can help you with that. Okay. So this is our last one. Like I said, after this lab, you will just have an assessment that you will be responsible for, and then that's it. You guys can enjoy summer vacation, and hopefully we will see each other next year. Okay. Thank you so much, and have a great night.